Hey guys, welcome to chapter 19. In this chapter we have three different topics. In the first topic we're going to talk about sexual reproduction, meiosis, and fertilization. So we're going to talk a little bit about another cell cycle that's a slight variation on mitosis. In topic 2 we're going to talk about Mendel and how genes are inherited. And in topic 3 we're going to talk about how we use all this information to do experimentation in the laboratory to help us learn a little bit more about human genes and how they work and what causes the diseases we've been studying all semester. So here's our topic outline. I expect a lot of this chapter to be pretty much review for you, so I'm going to go through it pretty quickly. But if you have any questions, please let me know about any of the items we're going to talk about. Here's our topic objectives. As always, these are what I expect you to have mastered before our next exam. And as always, if you have any questions or want to go through these in class, please let me know. Alright, so let's get started. First of all, before we get into meiosis, let's talk about the benefits of sexual reproduction. When an organism reproduces asexually, all they do is produce an offspring that's identical to the parents. This is just like mitosis, what we just talked about in chapter 18. Now this is fine if the environment is stable and it's not going to change, then the organism is going to be successful and is going to survive to reproduce. However, as we know, the environment doesn't always stay the same. And so what sexual reproduction allows for is for a reshuffling of the genes between different individuals. This can produce three different types of offspring. We could have one that's better suited for the environment. So if they inherited both these really strong traits from the parents, then this offspring may be a really strong offspring that could go on and continue to survive in this environment even if things changed. Or we'll have an offspring that's just as equally suited and will survive just fine. And then there's always the possibility that there will be an offspring that's not as well suited for the environment and is not likely to survive. And so it's really important to understand this, that this reshuffling of genes provides at least 2 out of 3 options that are going to help the species survive and go forward. This is also another aspect of it is it allows for harmful mutations to be moved from that population faster. And this is because of the fact that half the genes are being passed from one individual and half the genes are being passed from another. So if one person has a harmful mutation, chances of that offspring inheriting, that offspring can't inherit the full genome of that person. They will inherit half from their other parent, which will help minimize that mutation. So let's talk about the cells that are involved in sexual reproduction. First of all, I'm going to talk about sexual reproduction, meiosis. Everything is in diploid cells. Not all cells are diploid. It's important to understand this. Not all organisms are diploid. For instance, the strawberries that you guys smashed up in B201 lab were triploid. But we're going to pretend like everything's diploid because it's just easier for our brains to focus on that in this lecture. But diploid cells contain a full complement of the DNA, and in this case it's a 2x. As I said, if it's a triploid organism, they have 3x. The haploid cells contain half as much DNA. They have 1x to complement. So in humans, this is 23 chromosomes. And this is what we're talking about when we talk about your sperm and egg cells. The sperm and egg cells are referred to as germ cells or gametes, and these are the cells that specifically participate in sexual reproduction. And it's important, as I said, to note that that not everybody is diploid. There's all sorts of variations on this, but diploid is a pretty standard arrangement of chromosomes. So make sure you have these terms correct before we move on to how these haploid cells are actually produced. So the haploid cells are produced through a process known as meiosis, and you should be pretty familiar with this, but we're just going to go through it pretty briefly. Meiosis occurs in 2 stages, meiosis 1 and meiosis 2. These stages of meiosis 1 and meiosis 2 look a lot like mitosis on the surface, but it's not exactly the case. The chromosomes are being separated different, and that's really where the paramount issue or paramount situation comes from meiosis. So it's really important to understand that, and we'll walk through how these chromosomes are separated differently in the next couple slides. Also important is that meiosis only goes through one S phase, even though it goes through 2 divisions. This is how we get to half the DNA. And this process, whereas mitosis usually takes at most several hours, meiosis can take several decades. In women, their eggs undergo meiosis 1 in development and meiosis 2 when they hit puberty. So it's important they understand that this process can take a long time. So let's look at these chromosomes. Remember, we start out with, you originally have 2 chromosomes, one from your dad, one from your mom, and then these are duplicated into sister chromatids. So you end up with 4 at the moment. And this forms what is known as a bivalent, also a tetrad. You'll hear me refer to it as a tetrad throughout the lecture, but the 4 of them together, and they're all kind of clumped in this group like you see there. And so cells are not considered haploid until after meiosis 1, and that's when we're going to divide. And what you can see here on the second image on this slide is the difference. So in mitosis, during metaphase, the chromosomes line up on the metaphase plate, and as they divide, each cell is going to get one of each of those 4 chromosomes you can see there. Whereas in meiosis 1 at metaphase 1, the pairs are going to separate. So the M1 is going to go one way, the P2 is going to go one way, the M2 is going to go one way, the P1 is going to go one way. And this is why it's considered haploid, because even though they technically still have the same number of chromosomes at the end of meiosis 1 as mitosis, they're duplicated copies of the same chromosome. So they aren't the same thing as a diploid cell. So what else happens during this time with the chromosomes that is different than mitosis? And one of the big answers is genetic recombination. Now, we've talked about this process when we talked about homologous gene repair back in Unit 2, so review that if you need a little bit more on how this actually happens. But in meiosis, this process is known as crossing over. And what happens is the chromosomes are going to share and mix up DNA that is the same information. So they're going to always pass over the same genes. It's not going to mix up, the eye color gene on one chromosome for the hair color gene on another chromosome. They're always going to be swapping out the same things. And this creates the synaptomal complex and creates a chiasma. And what that does is it helps hold this bivalent or tetrad together. And that helps create this really strong structure that you see here, and we'll talk about how that divides here in a second. Most chromosomes do crossing over about 2 or 3 times across the whole thing, but it just depends. Now, an important aspect of this is that if there are genes that are all close together, they're likely to be crossed over together. If they're genes that are farther apart, they're likely to be mixed up more, and this is going to come into play when we talk about genetics in the next topic. So now that we know about crossing over and the bivalence and everything, let's look at how these chromosomes actually separate into one chromosome only. So we start out in the meiotic metaphase 1, and you can see the chiasmas here. And at this point, that kinetochore, that protein that's on the middle of the chromosome, acts as a single unit to keep the 2 sister chromatids together. And so in anaphase, the 2 sister chromatids are going to pull apart. And this is because the cohesions, the things that hold the chromosomes together at the ends, are loosened up, and so they can be separated, whereas the cohesions in the center of the chromosomes are still together, and that keeps those sister chromatids together. We then hit meiotic metaphase 2, and this is when the cohesions in the middle are going to separate. And this is when the kinetochores start acting on their own, and the microtubules are going to separate them on their own this time. But you can see once again still how that crossing over event has changed these. So each of these chromosomes is still a unique chromosome from what it was before. So here's a review of meiosis, and it compares mitosis here for you as well so that you can have a reference back to what we're talking about. As you can see, we have the 2 different meiosis steps. We got meiosis 1 and meiosis 2, and we have only one S phase though. So these cells, when we undergo meiosis, will generate 4 unique cells. These cells are very different from each other and from their parent cell. Whereas in mitosis, we have 2 daughter cells that are identical to the parent cell. And this is completed through crossing over. So make sure you understand this chart and how these work and the different processes here. Alright, the root of all this comes back to genetic variation. We already talked about that. Genetic variation is what allows species to survive in an ever-changing environment and allows evolution to move forward. And meiosis is how we get that genetic variation. There's 3 things that help these individual genetic variation come to be and how we all can be unique. And it's through sexual reproduction. The first in that you get half your chromosomes from your mom and half your chromosomes from your dad. So it doesn't matter if you look just like your dad, you still have half your chromosomes from your mom. And that makes you a unique individual. And even if, let's say that you have a ton of siblings, your siblings are never going to have the exact same genome as you unless you have an identical twin. And that's because of crossing over and independent assortment. So every time gametes are produced in either of your parents, the crossing over is going to occur. And that means that different chromosomes are going to be mixed up. Different genes are going to be mixed up in a different way than they were before. And that is unique every time. And so in women, since their eggs are kind of always there, this may not be a huge amount of gametes produced, but in sperm, we're talking millions of sperm being produced all the time. And so this random change all the time is going to produce a lot of genetic variation in those gametes. And then the last thing is the independent assortment of parental gametes. So not only is the crossing over mixing up the chromosomes, but you may get chromosome 1, 4, and 5 from your grandfather on your dad's side, whereas your brother may have gotten chromosomes 6, 7, and 8 from your grandfather, and then you guys got the rest from your grandmother. And so that's how those things mix up. They don't sort together, they always will sort independently. So it's just a random bag that you get in every single gamete as to what from the parents, from your grandparents actually, are being passed into those gametes that went together to make you. Now, meiosis isn't perfect, and we've seen this happen before, and we talk about this with Down syndrome and other diseases, but we have errors in meiosis. Sometimes the chromosomes fail to separate properly, and this creates what's known as non-disjunction. Now, if we're talking about a complete duplication of the chromosomes so that the gametes end up with twice as many, that's going to be considered that's going to be that is non-compatible with life. We have not seen humans survive with 3 copies of every chromosome, but we can see survival and decent lives from people who have one extra copy of chromosome 21, or there's a couple other genetic disorders out there where they have one copy of some of the smaller chromosomes. But the bigger the chromosome that's extra, the harder it's going to be for that person to survive successfully. We just have seen that the more extra genetic information is not necessarily better for people. And so let's wrap up with fertilization, and this is what gets us back to a diploid cell. So a zygote is a fertilized egg, and this is what happens when the sperm meets the egg, and you can see a picture of this here with all the different sperm. Now the first sperm that makes it through that outer shell, which is known as the zona pellucida, is going to trigger a calcium release, and this changes that outer shell to a really hard shell that the rest of the sperm can't get through. Once again, this prevents extra DNA from being incorporated into this new offspring. Now, if we have a non-disjunction event or extra sperm that make it into the cell, this is when we see those early term miscarriages. So before week 12, that's when a lot of miscarriages will happen, and that's because of some kind of genetic issue. Generally speaking, there's other things that can cause it, but in genetics, that's where we see that show up the most. And so that's how this fertilization process works. The sperm will digest a small part, get in there, changes the outside shell so nobody else can get in there, and then the process of fertilization is not complete until the egg and the sperm fuse their nuclei to create a new diploid cell. So this is the end of topic 1 in chapter 19. I know we went through meiosis pretty fast, but as I said, I expect most of it to be a review for you. Please let me know if you have any questions, and we'll go through it again in class if you need some more help.